Hi everyone, my name is Daniel Astudillo. I'm the head of marketing here at Recce Pharmaceuticals. We are ASX listed and Frankfurt listed. Got our usual disclaimer, and here we have our team. Our chairman is Dr. John Prendergast. Dr. Prendergast is also chairman of a New York Stock Exchange biotech company, as well as also a lead director of a NASDAQ listed biotech company. His doctorate is actually in the study of bacteria and blood diseases, and as you'll see further on, as an infectious disease company, that's actually quite important to us. As some of you already may know, James Graham's our CEO and Managing Director. By way of background, James was actually the founding investor of Recce and has been involved in almost every capital raise round to date. Alongside him is Michelle Delizia, who's our Chief Scientific Officer and co-inventor of Recce anti-infectives. So when we look to where that technology has actually transitioned to, we really consider from a position of an umbrella as an infectious disease company with two pillars, antibiotics and antivirals. Now the commonality between the two is, well, of course you should never use antibiotics on viruses, but it's really the binding and interaction with the protein of the cells. So the outer cell of the bacteria or the envelope of the virus itself. That's what we believe we interact and bind with. We currently have 2 clinical programs running in the background. Our first program is, of course, our sepsis-focused program at CMAC's Adelaide Research Facility as a phase 1 safety study in healthy males. And that's using our lead compound, Recce 327, as an intravenous solution. Parallel to that is our second clinical program, which is a topical burn wound infection program, really utilizing our technology as a spray-on antibiotic on the vulnerable burn wound itself. It's a phase 1/2 study out of Western Australia, led by burns experts and former Australian of the Year and global burns expert Professor Fiona Wood. Outside of that, we have the traditional preclinical programs, really moving them through the usual safety and efficacy type studies with the purpose of bringing them on as other clinical activities. This is really a pipeline as a traditional snapshot with further readouts in the coming months. So really our lead indication, sepsis. What is sepsis? It's blood poisoning septicemia. It's the number 1 most condition treated in health, double the second most. It's a major unmet medical need. There we see about 50 million persons each year contract sepsis, and 11 million of those in fact actually die, or in the hospital it's a 1 in 3 death rate. And the reason that is that there is no antibiotic specifically for the treatment of sepsis. So why is that? What's the clinical challenge here? The challenge is the treatment paradigm. When a patient presents to the hospital showing signs of sepsis, the clinician quickly draws blood, rushes it off to the lab, where they cultivate that blood out to try and determine, 1, what type of bacteria is in that patient's blood, and what antibiotic that bacteria is susceptible to. Now that takes time. For every hour left untreated, the likelihood of survival decreases by 8% each hour. So our antibiotic is actually designed with the end in mind to avoid that time-consuming guesswork, which current practice is to really administer a cocktail of antibiotics in pure hope that the bacteria actually responds. So the faster you get into the blood system, the likely the better patient outcome. And we believe we've eliminated that guesswork, and of course, getting on top of patient infections leads to better patient outcomes. All existing antibiotics are naturally derived. You take the bacteria or the fungi, you cultivate them out, and essentially you're putting them against that antibiotic, against the offending bacterial fungi. But at the end of the day, you're only as good as what's found in nature. And that bacteria will continue to mutate, which it always has and it always will. This is essentially a survival mechanism. And that traditional mechanism no longer works. Beginning with the end in mind, we've designed ours to have multiple mechanisms of actions, and particularly to be attracted to gram-positive and gram-negative. And it doesn't discern whether or not they try to mutate to superbugs. It's bacteria to us at the end of the day. This is really talking a little bit about that. Basically, we have an initial attraction and binding to the bacteria itself, whether gram-positive or gram-negative. In this case, we're looking actually at E. coli. We enter or we permeabilize the bacterial cell wall, and when in the cell itself, we interact with the ATP and the ATP production. And that ATP production is actually effectively the beating heart or the lifeblood of the bacteria itself. We shut down that beating heart, and we do it, and we shut it down irreversibly. That's the first mechanism. The second mechanism is that we stop the cell from dividing or the infection from actually spreading. And as you can see in stage 4, we know that in high concentrations we take advantage of that metabolic process, which creates that intercellular pressure to really cause that bacteria to burst. No other cells in that body have that high internal pressure, and that gives us a high degree of selectivity amongst the other activities happening here. This is really looking at it at a molecular level. It's all well and fine to actually animate it, but once you see the real images, it certainly is something else. Now the question is, does it work and keep on working with repeated use? The simple answer is yes. It's a fundamental challenge of all existing antibiotics to date. And here the commercial antibiotic being amoxicillin, which generates over US $10 billion in revenue, stops working only after a number of repeats. We've actually extended that out to over 100 repeats, but for this sake it's 25. Next is our phase 1 human clinical trial, which, as I mentioned, it's focused on sepsis. It's a safety study, meaning that people are healthy, and it's really a dose escalation study on really how much of the compound can a person reasonably take in the blood. We've obviously done that many times in animals, and we seek to purposely find that ceiling with the intention of that therapeutic window between where it works against the bacteria and where there's too much compound administered into the blood. We've cleared up to 500 milligrams up to cohort 3, and dosing will start sometime this week for the 1,000 milligrams, so it's really exciting there. Obviously, parallel to that, as I mentioned, we have some further exciting data to come out of our topical phase 1/2 clinical trial. Again, this is sponsored by the WA Health Department. Really doesn't cost us anything. It's caveat-free in regards to the clinical data supporting our overall development. This really here are the different types of bacteria we've actually shown to be efficacious against in the phase 1, and we've seen a visible reduction in the infection within less than 24 hours. To which the actual investigators have actually recommended to reduce the treatment window by half, as it was evident that all infections were cleared after their respective time. In the background, we're also working with Murdoch Children's Research Institute. One example is utilising our new class of synthetic anti-infectives as an oral solution targeting H. pylori, which really exists in the upper duodenum of the gastrointestinal tract. People often say, what about the good bacteria? Well, they exist in the lower gut or the lower intestine. And as our data today supports, or really indicates, I should say, we're 100% water soluble across all pHs. It breaks down as it interacts with the bad bacteria in the upper duodenum, or it breaks down due to that solubility. So when we get to that good bacteria, we don't appear to have any of that existing antibiotic properties, which is a unique advantage in our case. This is really looking at our antiviral programs. Just realize I'm looking for time. Next, we have our intellectual property. We have patents. We have a broad range of patents across the world. We also have a qualified infectious disease product designation from the US FDA, which is government-backed. And that government-backed market exclusivity comes above and beyond our patents, which would start from time of approval. Now, patents can fall over, not at all concerned about anything there, but if they did, we have that government-backed exclusivity unique to that sepsis program, starting only from time of approval. So our manufacturing, we have a manufacturing facility in Macquarie Park. We don't own the building, but we own everything inside it. It's cheap to make, and we're currently producing in volumes to planning phase 1 and 2 clinical trials. Really, I just want to finish off here. We're well funded with approximately $15 million of bank, just under 5,000 shareholders. We have a low fixed monthly burn rate between $150,000 to $200,000 a month, and we have the first new class of antibiotics in over 30 years. We're an infectious disease company in the midst of an infectious disease crisis, and we very much look forward to communicate news flow and opportunities as it presents in the company along. Thank you so much for your time.